Hello dear students, this is Dr. Maushami Das and I am going to deal with you in this agricultural microbiology course the topic which is deal with the three important terminologies which you should understand, that is rhizoplane and phyllosphere and spermosphere. These are the three important terms I am going to share with you today, some little overview. Now coming to the first important word or term which is called rhizoplane. What do you understand by this word rhizoplane? The term mainly coined by Clark in the year 1979, and this rhizoplane is mainly the external surface of the plant root. For 1 marks MCQ it can come, so just have a look in this particular terminology. Apart from that, if I talk about some properties of this rhizoplane region, this is the direct region with the contact of root and partly it is a converted region too. With that, it is having a influential activity of bacteria and fungi in this particular region. Now, if I talk about the community, the microbial community which is present in this particular rhizoplane, as you can see here, this blue lines, this is particular the zone which is called as rhizoplane, and the surrounding location of the microbe, it is available actually for the bacteria, it is 10 to the power 5 to 10 to the power 7 CFU per ml, and largely the gamma and alpha proteobacteria are more prevalent here. For 1 marks MCQ, this is a very important part, which type of bacteria is more prevalent in the rhizoplane. So you will say that the gamma and alpha proteobacteria are mainly profusely growing in this particular rhizoplane region. Now, if I talk about certain genera which you need to understand of the scientific name, like Micrococcus, like Desalphovibrio, Vibrio, these are the typical group of bacteria you can come across in this particular rhizoplane region. Now, importance, why we should study the rhizoplane? The rhizoplane community study is because to see the growth of the plant and as well as the harmful microbes availability and how to inhibit them. To know this particular factor, we generally go for the rhizoplane. This much is enough to know for you for right now. Now, phyllosphere, phyllo, this word means leaf. Sphere means the region in and around the leaf or on the surface of the leaf, the region which prevails and the community of the microbe which be available in this particular region, that is called as phyllosphere. Now talk about the phyllosphere, mainly the important microorganism play a role in this zone and they are very different from the rhizosphere, and this zone is dynamic zone because they are an transient zone too, because here always you cannot find the same type of microorganism and they are not resident flora. Resident flora means the permanent inhabitant of that particular place, but they are the transient flora. Due to the fluctuation of the spore shift or due to the shifting of the different type of spores from one place to another, you cannot find for a long duration of time the availability of same type of microbes in this phyllosphere region. Apart from that, certain plant disease management, plant growth, nutrient acquisition, and with that the protection to the plant, these all activities will be performed in this particular region of soil. Now the community or the microbiota which we generally come across in the phyllosphere. But you can see at your right side picture, you can see the groups of the microorganism. You see this particular tree and here the leaves where you can have highlighted by the different colors the availability of bacteria, fungi, then virus, protist, algae and nematodes. If you see the zones wherever we talk about the phyllosphere, there one is the colosphere, that means the stem part, the phyllosphere, that means the leaf part, leaf lamina, leaf midrib, leaf vein, these are all the palm of the leaf. These are all the regions where you can find the availability of microbe. You can see also in this picture, this red color is depicting the harmful microbe and the black color is depicting the beneficial microbe. So in the regions like in the seed or in the regions like in the carposphere, you can have the availability of beneficial microbes too. So both the types of microbe, beneficial and harmful, are there in these different region like colosphere, anthosphere, spermosphere or in this phyllosphere region. Few examples which you need to know under the main 4 families which are dominating in the phyllosphere region, that is proteobacteria, firmicutes. Firmicutes is a type of a actinobacteria. Apart from that, bacterioids and a large group of actinobacteria or endophytic bacteria you have studied in your microbial ecology self study. So these particular type of microorganisms are prevailing here. Now few common genera which you should understand as a name, it can come for 1 marks MCQ, like Methylobium, Hypomicrobium, Methyloblia cella, Actinobacteria, Macilla, Flavobacterium, Pseudomonas, and also the Rathylibacter. These are different groups of microorganisms which you can find here. Cyanobacteria is not so dominant colonizer, they are the frequent colonizers because they need certain water, thin film of water. Yeast in that you can find a good species, that is Cryptococcus and Rhodotorula and Sporobolomyces. These are the different yeast genera. If you talk about the count or the intensity of the colony, that is 10 to the power minus 2 or 10 to the power 8 CFU per gram of filamentous fungi will be available. In that the prominent genera in this Phyllosphere region you can find Penicillium, Cladosporium, Alternaria, Acrimonium, Mucor and Aspergillus. So you need to remember the names of the different group of fungi, bacteria and actinomycetes which are prevailing in this particular Phyllosphere region. Now why this availability is so pronounced, or they are available in this particular Phyllosphere region? What can be the reason? The reason is because of the different type of exudates which is released in this region. For example, the types of the amino acids, the types of the organic acids or the types of the vitamins which are released. Because of that, the more nutritional availability is found in this Phyllosphere, but they are transient. Once again I say you, they are not the permanent resident flora because they can shift, they can replace from one place to another. Now spermosphere, what do you understand by this terminology called spermosphere? Sperm, that means the reproductive unit. So spermosphere means, you can see here at your right side, this is actually a seed. In that seed you can see the regions outside and inside the layer, where you can see in between the soil or in the seed borne microbial community, especially in the germinated seed, the availability of different group of microorganism. If you see this picture, what you can see here, one is the endosperm, then this is the bran part of this seed, then this is the safe spermosphere region, and this is the germ cells. So these zones which are there, depending on the nutritional composition and pH and moisture, the difference of the microorganism can be found in this spermosphere effect. You can see the blue colors. What is depicting in this picture, blue color? It is the aerobic microflora, whereas the anaerobic microflora is mainly found in this germ part and also in the safe spermosphere part. So likewise you can have the difference of the community of microorganism based on the presence of the oxygen or intensity of presence of the oxygen. Sometimes microaerophilic microbes are also available in the just germinating section. Now Nelson, the scientist, has defined this spermosphere as the zone of the microbial interaction around the seed and which is under the influence of the seed carbon deposition. This particular definition you need to remember whenever there will be the question of define or elaborate or conceptualize the term called as spermosphere. Now the marking parts, if you see here in this crucial zone, where you can see the high contact between the plants and the pathogens as well as the soil microorganism, they also having a sometimes deleterious effect and sometimes the nutritional effect too. Small quantity of release of exudates are there because it is the endosperm gland region, so there must be some exudations also take place. So due to that, there will be the short transient process of these exudations with a heavy complex community. Studying of the spermosphere is not so elevated till now because of many factors. The reasons can be, for example, passive exudation process, for example, the complex community presence, and also they are transient. Because of these reasons, the total detailing study of the spermosphere microflora is not so flexible like the rhizosphere. Now in this particular picture, what I make to you to understand, that is this particular region, this seed region, around that seed region, if you see these strangle parts are nothing but the passive exudation process, and these circular parts or circular labels are basically depicting you the microbes which are adapted due to this exudation in and around this region. And you can see here based on the spermosphere effect, the characterization or characteristic property, if you see this, the exudation of the seed having the three peak stages are there of this exudation in this spermosphere. One is the seed imbibition stage, that means when the seed takes the water, they swell up, that is called the seed imbibition stage, that is the first peak. That time, after the few hours of imbibition, you can see the radical get more pronounced. After this, you can see the carbohydrate, organic acid, alcohol, fatty acid, amino acids, they are getting more released here. Then these exudates of seed which is having a high alcohol or fatty acid or such type of compound, they affect the pH and the soil moisture, even the temperature. There are also certain analytical methods by which you can also study this seed exudates characteristic features, and you can also predict the group of the microorganisms availability in such type of areas. Now coming to the another analytical techniques like TLC, HPLC, you can go for ion exchange chromatography, GC. These are certain processes by which you can study the types of the compounds which are getting released as exudate in the spermosphere. Apart from that, there are also certain other two regions in this spermosphere. One is the host dependent region and another one is the soil dependent region. So depending on the availability of these regions, you can have the diversity of the soil microorganism like eubacteria or gram negative or positive bacteria, or certain true fungi like for example ascomycota, or for example the oomycetes. These are the certain group of fungi which you can see here in this particular spermosphere region. Now this is also you can study by flame or PLFA or CLPP. These are the certain methods by which you can study them. This is the chart, this is the elaborative chart which you see in this particular PPT, where at the left side you can see the different type of compounds starting from carbohydrate, amino acid, organic acid, alcohol, certain secondary metabolite, fatty acid and proteins, and how they can be analyzed by this different type of technique. Majority of the techniques are the chromatographic techniques or mass spec, जैसे GCMS, NMR, HPLC, TLC, RPHLC, electrophoresis, immunoblotting. These high analytical throughput procedures are available to detect this type of characteristic of compounds which are present in the spermosphere region. Now, as I was telling that spermosphere is also a very good region for the different group of microorganism, but depending on the host dependent region and also in the region where the microbial community actually decide, here you can see the germinated medium, that is also an influential region, and also the bacteria and fungi are more dominating in this particular spermosphere. Type to type plants also differ. Sometimes there is a cereal plant, sometimes it is a plant which are having the nuts. So depending on cereal grains or nuts or types of the vegetation of plant, you can see the spermosphere is having a diverse or dynamic community of the microbe, that you need to understand. Apart from that, if you see the types of the microbes at your left side, starting from Azotobacter, Bacillus, Streptomyces, or if you talk about the fungi, Fusarium, Trichoderma, then Methylobacterium as Proteobacteria, and sometimes a mixed populations like for example Trichoderma harzianum and Pseudomonas fluorescens can also found to be grow in this particular spermosphere region, and they have individually their important roles like Azotobacter or Bacillus. Such type of microbes are mainly promoting the germinations of the seed, also the root volume. Sometimes there are also certain microorganisms which are helping in the increase in the germination process like Trichoderma aestivum, or suppressing certain harmful fungi or bacterial effect, or sometimes they are also helping in certain plants like chickpea, pea, soyabean, lentil, tomato in the formation of the germination and exposition of the biotic stress. So such type of different activities or role are played by these bacteria, fungi and actinomycetes in spermosphere. So with that I would like to conclude some questions for this particular chapter, like comment in brief the role of the microorganism found in the spermosphere. It can come for 5 marks, or the question can come like elaborate or discuss in detail about the different microbial community present in Phyllosphere, Spermosphere and in the rhizoplane. Or simply by 3 marks question if it asks like what do you understand by the rhizoplane community, or what do you understand by the term rhizoplane, or differentiate between Phyllosphere and Spermosphere microbial community. What are the different type of exudate which are released in the spermosphere, Phyllosphere and rhizoplane? Such type of questions can be asked for short marks, and even all together these questions can be asked in the 7 marks. And apart from that, for 1 marks MCQ also special words or examples or terminologies can be asked. So this way I have concluded the particular chapter where I have dealt with the 3 important terminologies, that is rhizoplane, phylloplane and also this particular phyllosphere and this particular zone of the microbial community discussed in this particular topic. Thank you for your patience hearing.